Good morning, everyone. My name is Alireza Haghighi. I'm the founding director of the International Center for Genetic Disease and an assistant professor of medicine at Brigham and Women's Hospital and Harvard Medical School. It's my great pleasure to welcome you to our third lecture of the Keynote series on genomic medicine, which is organized by the BWH International Center for Genetic Disease, or ICGD. In this series, we feature talks by internationally renowned scientists as they explore the very foundations of life and improve our understanding of genetics and biology. The series is scheduled for the third Tuesday of each month from 9 to 10 a.m. Eastern Time, and it's on Zoom. Today, we are delighted to be joined by Dr. Robert Higgins, president of Brigham and Women's Hospital and a board member of ICGD, who will provide opening remarks. And of course, we are also pleased to host Dr. Huda Zoghbi as our keynote speaker, who I will introduce separately. Today, our audience includes participants from 43 countries and 5 continents. Dr. Higgins is the president of Brigham and Women's Hospital and executive vice president at Mass General Brigham. He previously served as surgeon-in-chief at Johns Hopkins Hospitals, the William Stewart Holstead Professor of Surgery, and director of the Department of Surgery at Johns Hopkins School of Medicine. He's a leading authority in heart and lung transplantation, minimally invasive cardiac surgery, and mechanical circulatory support. Dr. Higgins has served in numerous national professional leadership roles, including president of the Society of Thoracic Surgeons, president of the United Network for Organ Sharing, president of the Society of Black Academic Surgeons, and as a member of the board of directors of the American Board of Thoracic Surgery. Dr. Higgins received his medical degree from Yale and completed a residency in general surgery and served as chief resident at the University Hospitals of Pittsburgh. He earned a master's degree in health services administration at Virginia Commonwealth University. My colleagues and I at the International Center for Genetic Disease are grateful to Dr. Higgins for his strong support of ICGD and its mission to extend the benefits of genomic medicine worldwide. Without further ado, I welcome Dr. Higgins to deliver his remarks. Dr. Higgins.
Thank you. It's a great pleasure to welcome you to this third lecture in the annual Keynote series on genomic medicine sponsored by the Brigham and Women's International Center for Genetic Disease. The center is based at the Brigham and Women's Hospital, but I'm pleased to note that it reaches far beyond that to our Harvard-affiliated teaching hospitals, including the Mass General, Boston Children's, Beth Israel, Deaconess, and the Dana-Farber, as well as to the Broad Institute of MIT and Harvard and the School of Dental Medicine. Today, it's a pleasure for us to host scientists, clinicians, and many others from countries around the world. Before Alireza, the founding director, introduces today's distinguished speaker, Dr. Huda Zoghbi from Baylor College of Medicine and the Howard Hughes Medical Institute, I just want to note briefly why genomic medicine, as I'm learning and being educated, is such an important emerging field in contemporary medicine of today. As defined by the NIH, genomic medicine is the emerging medical discipline that involves using genetic information about an individual as part of their clinical care. As a clinician, this is very important to me, and I'm sure many others. And it also speaks to the health outcomes and policy implications of that clinical use. Its clinical implications are rapidly expanding, and these include diagnostic testing, pharmacogenetic testing, risk prediction using polygenic risk scores, population screening. The list goes on to identify novel therapeutics and interventions, such as gene editing in this process. Over the past decade, genomic medicine has gone from a mere concept to an actual clinical implementation. I'm proud to say that President Obama's mandate at the 2015 State of the Union called for a federally funded large-scale precision medicine initiative, now called All of Us, and specifically emphasized this national commitment to genomic medicine as a new paradigm. All of you know that genomic medicine provides a route to translate innovative science, ranging from novel diagnostics and effective therapeutics, to enhance patient care and the prevention of disease. We, as an internationally recognized academic medical institution, seek not only to provide the best care for our patients through cutting-edge genetic science and research, but also to promote genomic medicine worldwide. This center focuses on the analysis of patients and healthy subjects from different parts of the world to elucidate the genetic causes, consequences, and create prevention strategies and treatments of disease. Currently, it will interest you to know that close to 80% of the genomic data is limited to individuals of European ancestry. The practice of genomic medicine is also only just beginning to be implemented in many parts of the world. So we hope that the research, clinical work, and capacity building projects of the International Center for Genetic Disease will have a significant impact in eliminating these disparities. So I'll finally state that we are committed to address representation and diversity in genomics. We need genomic and clinical data from thousands of individuals from diverse populations in order to understand the genes and changes in the DNA sequence that lead to human disease phenotypes. I'm proud to say that the center provides an infrastructure to compile and analyze genetic knowledge across geographic borders and uses cutting-edge capacities and capabilities to improve health in both international and local settings. I'm looking forward to today's lecture, Therapeutic Approaches to Tackle Neurodevelopmental Disorders, Lessons and Prospects by Dr. Zoghbi, which will provide, I'm sure, a state-of-the-art example of the enormous progress being made by researchers in using genetic information to understand debilitating human disease. Thank you again for the opportunity to spend time with you, and I look forward to the lecture today, and I turn it back over to Dr. Haghighi.
Thank you so much, Dr. Higgins. We are thankful for your generous support to the center. Today we are delighted to host Professor Huda Zoghbi as our keynote speaker. Dr. Zoghbi is Distinguished Service Professor of Pediatrics, Molecular and Human Genetics, Neurology, and Neuroscience at Baylor College of Medicine. She is also an investigator with the Howard Hughes Medical Institute and the founding director of the Jan and Dan Duncan Neurological Research Institute at Texas Children's Hospital. Huda was born and raised in Beirut, Lebanon. She attended American University of Beirut Medical School for 1 year, but due to the Civil War, she moved to the US and received her medical degree from Mohari Medical College. She joined Baylor College of Medicine for her residency and subsequently obtained additional training in molecular genetics. Dr. Zoghbi's expertise ranges from neurodevelopment to neurodegeneration. She and Harry Orr discovered that spinocerebellar ataxia type 1 is caused by expansion of a polyglutamine tract, and her subsequent studies further demonstrated that such expansion leads to accumulation of the mutant protein in neurons. These findings have had profound ramifications since many late-onset neurological disorders involve similar accumulations of disease-driving proteins. Her work in neurodevelopment led to the discovery that gene MATH1 governs the development of several components of the proprioceptive, balance, hearing, vestibular, and breathing pathways. She also discovered that mutations in MECP2 cause the postnatal neurological disorder Rett syndrome and revealed the importance of this gene for various neuropsychiatric features. Dr. Zoghbi has trained over 100 scientists and physician scientists. She has been elected to the National Academy of Medicine, the National Academy of Sciences, and the American Academy of Arts and Sciences. Dr. Zoghbi's honors include the Greengard Prize from Rockefeller University, the Shaw Prize in Life Sciences, and Medicine, the Breakthrough Prize in Life Sciences, the Canada Gardner International Prize, and the Victor McCusick Leadership Award from the American Society of Human Genetics, among others. She has also received honorary degrees from Harvard University, the University of Massachusetts, and Yale University. It's an outstanding honor to welcome Dr. Huda Zoghbi as our keynote speaker, who is going to talk about genetic and therapeutic approaches to tackle neurodevelopmental disorders. Huda?
Thank you, Alireza. It's truly an honor to be here. And thank you, Dr. Higgins, for sharing with us all the wonderful work that you're doing at the hospital. I will now share my screen and hopefully everything will work. So, can you see my slides in full? Excellent. So today I would like to share with you my journey to try to understand better neurodevelopmental disorders. And this is very fitting since April is the month for recognizing autism. And what I would like to do is really, although I will focus mostly on one disease, Rett syndrome, I will go back and forth and share examples as what we've learned from Rett syndrome and other disorders, and how could this be applicable to many other neurodevelopmental disorders. So I think many of you might have heard, I started my career as a physician, as a child neurologist, and it was actually encountering patients with neurological disorders that inspired me to go into science. And it is in particular the first inspiration with this young girl, Ashley, who was a healthy child. You see her here sitting straight, reading a book. You see her here riding a horse. She was able to do so much by 2 years of age, including singing nursery rhymes. And then all that was lost after 2 years of age. She stopped greeting her father at the door, stopped talking, stopped using her hands, which she used, as you can tell, very well early on. And you see her in this picture, wringing her hands. So having seen her, I was really intrigued. How could a disease progress so quickly without being degenerative? And we knew it's not degenerative because right when I saw Ashley, Ben Hagbert has published a paper in the Annals of Neurology describing patients from Europe who had Rett syndrome, and it was clear that all 35 cases did not have a degenerative phenotype. They were born healthy and went through that course. And he honored he named the syndrome Rett syndrome in honor of Rett, who described it in the German language in 1966. And it was actually serendipity that I saw another girl at the same, within a week, with Rett syndrome. And here I just want to show you a video to see really what Rett syndrome looks like, so that you can recognize it affects all children of all ethnicities. And you can see here rocking activity, autistic features, hand wringing, and then you'll see the challenge when she's trying to move. She has the motor strength to walk, but she can't walk. It is an apraxia, an ability to execute on emotion. And then when she walks, she's very unsteady and falls backward. So having seen 2 girls within a week, I was really intrigued and started looking for more and identified a few cases. At the time, no one really has known about this syndrome yet in the United States, but the challenge was all of them were sporadic. So you see here, just one in a family, all females. And I decided to go into research to find the Rett syndrome gene. And as you can imagine, in 1985, finding the gene for a sporadic disease wasn't quite possible. So my mentor, Art Boudet, said, pick a Mendelian disorder. And that's why I picked the spinocerebellar ataxia that you've heard Alireza describe our work there. And it took actually a while. It took 16 years from seeing Ashley to find the Rett syndrome gene, because we really had to do it brute force, marching down the X chromosome gene by gene until we found the right gene. And this was discovered by Ruthie Amir as the gene encoding methylcytosine binding protein 2. This is a protein that was first described in 1990, early 90s by Adrian Bird as a protein that bind methylated cytosine. That's the modified cytosine base pair. And we found mutations in this gene in Rett syndrome. So this was one of those first, if you will, sporadic diseases where now you know it is caused by a genetic disorder. And since then, we know so many more sporadic autisms and intellectual disability caused by new mutations in a gene. I put here some examples, but the truth is we're up to almost 1,000 and we're not finished. There's more to be discovered yet. So for the students and everyone else on this call, what took us 16 years today can be done in one day. So that's really important. And it is challenging to see children with developmental disability not knowing the cause. So we really have to do our best to do this and to reach all children, not just children with the resources to do this. So I hope this will continue to happen. And just to Dr. Higgins' points, we've realized here in Houston, where we've made many, many genetic disease discovery, is that we typically have much higher rate in people with European descent. And this is why we started what we call the Texome Project, so that we can reach people from underrepresented group who don't have the resources. And we would love to work with you and collaborate to continue to reach as many people around the world to find the remaining causes of autism and IDD. So what happens after finding a gene? In the case of Rett syndrome, because it's on the X chromosome, there's half of the cells having normal amount of the protein, and the half cells that you see on this cartoon as blank lack the protein because of the mutation. And when you have this mosaic pattern and a mutation that's inactivate the protein, you got the classic Rett syndrome, which you've seen in the video, affecting all parts of the nervous system, causing seizures, even causing Parkinsonism. Again, even though there's no degeneration, there's dysfunction of the cholinergic neuron. But once you have a gene, you start broadening the studies. And we've learned now that males, although we have seen very rare cases of males in families when we're trying to clone the gene, we now know that there are males, and when the male lacks a gene, because the male has a single X chromosome, it's a very severe phenotype. They typically have neonatal onset encephalopathy, motor problems, autonomic problems, and they die, unfortunately, by 2 years of life. But what we're also learning is that they're milder mutations. These are mutations that the protein is still made, either it's at lower levels or it has decreased function. And when you have these very mild mutations, what we've learned is that males can survive with these milder mutations. And some of them will present with learning disability and autism, or any one of these other psychiatric features. It could be anxiety, it could be OCD, hyperactivity, bipolar disorder, even juvenile schizophrenia. Obviously, this is not isolated psychiatric features. It's always layered on learning disability, but those are in different colors because each patient might present with a different symptom. So it's really has been quite informative to study the variant mutations in this gene and to learn that even a male can survive into adulthood if he retains the majority of the function of the protein, but partially loses it. And typically the mother who might carry such a mutation, if she's a mosaic, she will be fine. So this is really telling us about the exquisite sensitivity of this protein, of the brain to this protein. We're fortunate that mouse models replicate the features of this disease. You see here a healthy mouse, and you see the paws apart, and then you'll see the Rett mouse model where you see even this abnormal repetitive movement that you saw in the girl with hand wringing can be reproduced in the mice, and you see a little bit of a tremor here. The mice really replicate all features of the disease, and many mouse models have been generated, including many great ones by Adrian Bird and others that are in use. So having learned that we have a good mouse model, the first experiment we wanted to do is add back the normal gene. And we did this genetically first. We added a human gene that had all the regulatory elements of MACP2. So the mouse has its mouse MACP2, and we added the human MACP2. And to our surprise, we learned that doubling this protein also resulted in a neurological progressive disorder. In the mice, we saw features of autism, anxiety, learning issues, stereotype behavior, epilepsy, and these mice died by 1 year of age. This was really surprising to us. We did not expect this. And when we normalized the protein by breeding these mice to mice that lack the gene, the mice got better again. So this suggested to us there may be patients with duplication spanning this gene. And indeed, Linda Van Esch reported on the first series of these MACP2 duplication a year later, where she identified males that have duplications spanning MACP2. Their duplications are of varying size, but always MACP2 is in the minimal region of overlap. And they had all the same features we've seen in the mice, including premature lethality. And what we think is the majority of these features are actually due to MACP2 duplication and not many of the other genes in the duplicated region, because as I mentioned to you, the mouse share all these features. So I'm going to get back to this disorder later as we discuss therapy. But what we now can think of is, why is it that you have doubling of the protein and you have a problem? And what we learned is that loss or doubling molecularly are opposite of each other. So if you lose the protein, many gene expression changes happen. And if you gain the protein, the opposite happens. So what this told us, that this duplication is just doing too much of what MACP2 normally does. So you have to have MACP2 just right here at 100%. If you have none, it's fatal. If you have it in 50% of the cell, it goes as red. If you have 50% in every cell, we saw in mice, it gives milder disease. And if you have one of those mild mutations, this is one of those mutations that causes many psychiatric features. Now you get neuropsychiatric phenotypes layered on learning disability, and doubling causes a human and mouse disorder. And we found both in mice and humans that triplications are even more severe and fatal. So it is really important to keep this protein in check. And it is actually, when we studied this duplication, I started thinking, is MACP2 the only dosage sensitive proteins, or are there other proteins that's so dosage sensitive? And decided to test this hypothesis on a protein called SHANK3, where it was discovered first. The syndrome was reported by Phil and McDermott, but many mutations have been shown in SHANK3 and loss of one copy to cause Phil and McDermott syndrome, and point mutation to cause autism and intellectual disabilities, and rare cases of schizophrenia. However, there were duplications spanning the gene plus 30 additional genes. So we wanted to know if doubling SHANK3 by itself could be problematic. And these are mice generated by Ki-Hoon Han and characterized by him and Jimmy Holder. And if you were to watch this video, all you have to do is just look carefully and you'll see there's one mouse, this particular one, who simply cannot be still, constantly on the move, constantly on the go. And initially, we thought this is maybe a hyperactivity phenotype. We measured the activity and we see both male and female mice have this hyperactivity phenotype. But then we learned that actually it doesn't respond to amphetamine and it gets worse. And this led us to suspect maybe it's mania-like phenotype. Of course, you can diagnose mania in mice without human data. So we started back from the mouse, we went to humans and tried to see if we can find duplications spanning SHANK3 gene. And we identified a few patients that have duplication that span the isolated gene without the other neighboring genes. The ACR is a sperm-only proteins expressed in sperm. And here's an example of a 35-year-old with this duplication who had seizures and a diagnosis of bipolar, mood swing, anger issues, and attempted suicide. And the mice had many features that are reminiscent of mania that you see: lack of sleep, constant activity, and so on. But having seen seizures in this human, that sent us back to the lab, and collaborating with Jean-Rong Tang, we performed in vivo EEGs, and you'll see here the hyperexcitability in these mice and the electrophic seizures. And we pinned down to increase in excitatory synapses and identified the molecular mechanism for that. We also learned that in this particular form of bipolar, it's valproate and not lithium. Jimmy Holder tested various treatment, and it was valproate that helped these patients, this particular model. So this comes back to the point that Dr. Higgins raised, that identifying the genetic cause of the disease can actually guide the choice of the therapy. So to us then, we learned that what we've seen with MECP2 applies to other diseases. Here's, for example, SHANK3, loss of one copy causes Phelan-McDermott, triplication adding 50% more causes bipolar, and we've also studied a very fine hypomorphic mutation that causes isolated autism. So what we've seen with MECP2, we've seen with this disease. But it goes beyond that. This dialogue between mouse studies and human studies, I have discovered for the last few years, goes beyond just neurodevelopmental disorders, even to neurodegenerative disorders. So really, these mouse studies can help us predict candidate disease genes. But then if we go back to the humans that we identify with these diseases, we see additional phenotypes. We can go back into the animal and refine them, as I showed you with the seizures and SHANK3. And I just want to show you a few examples from our lab, where we found a negative regulator of the ataxin-1 protein, PUMIL-U1, its haploinsufficiency in mice, increased ataxin-1 and caused neurodegeneration ataxia. And this led us to discover that mild mutations in adult can cause ataxia, whereas haploinsufficiency, which is much more severe than the missense mutation, can cause developmental delay, ataxia, and seizures. We also found an interactor of ataxin-1 that haploinsufficiency causes neurodevelopmental phenotypes in the mice, and this led us to discover humans that we now call Kapicua haploinsufficiency syndrome. And Jihong Song from Harvard, together with Rudy Tanzi, collaborated with us, and they discovered that loss of ataxin-1 potentiate Alzheimer pathogenesis by elevating cerebral base 1. So the point is, and they've seen patients with copy number variants affecting ataxin-1 and have features of Alzheimer. So the point that this dialogue between mice and humans can be extremely helpful to solve disease, to understand mechanistic disease, and to even predict disease. So I just want to end up on this nuanced, important amount of the protein by sharing with you very briefly that we wanted to understand the regulation of MECP2, given how much its levels matter. So there's what we call cis-regulatory elements, or CRE, that typically bring transcription factor to regulate the expression of a gene. And Ying-Yang Shao in my lab identified such elements, and she found the deletion of one of those elements reduced MECP2 to 70%. So rather than having the normal 100%, now we have 70%, both in mice and human neurons. And what she found is in the mice, when you only reduce the protein by 30%, you get hyperactivity, anxiety, and social deficit, just a subset of the full-blown spectrum of the phenotype we see in rat mice. On the other hand, she identified another regulatory element that when she deleted, she saw an increase in the protein by 50%. And these mice had learning and anxiety and hyperactivity, again, a subset of what we see in mice that have 100% increase the MECP2 duplication mice. This is really important because it's going to tell you they're going to be patient with milder mutation that have these subset of phenotypes. But it even also tell you if we treat these diseases, even if we don't get back to the normal 100%, even if we move the needle to where we bring up some protein function back, we're going to rescue many features, and the same is true as we decrease the mutant, as we decrease the duplication protein. So now we can add, in addition to the rat and the severe duplication and loss of function phenotype, these mild and moderate features we see by just a 30% decrease or 50% increase. And I would like to venture to say they're going to be changes in the 10% range that might give you just late onset isolated psychiatric phenotype. And again, I don't believe this is going to be unique for MECP2. We should really think about it in the context of many of the disease genes that we know either loss or duplication can cause neurological phenotype. Now, moving from studying it at the molecular level, I just want to share a little bit with you about what we've learned about the function in the neurons. These are the features of Rett syndrome. It essentially affects all parts of the nervous system, central and peripheral and autonomic. And when we did knockout in different brain regions or different cell types in the mice, we discovered that certain neurons mediate some of these features. For example, hypothalamic neurons can mediate feeding abnormalities. They can mediate aggressive behavior. And dopaminergic neurons, some motor problems, and so on. Of course, GABAergic and glutamatergic, being the majority of the neurons in the nervous system, had many of the features. But the lesson we learned from this is that every time you take MECP2 from a neuron, you disable that neuron and you decrease its function by about 30%. So it functions, but it is functioning at lower capacity. And because we discovered that we need the gene in all the neurons to really have perfect function, eventually the system will crash if you only bring it back in inhibitory neurons or only bringing back in the excitatory neurons. We wanted to see if circuit manipulation will help. Would it be possible to, instead of thinking of just bringing the gene in one type of neuron or using molecular therapies or a drug, where we cannot find a drug that can activate all neurons, would it be possible to just focus on one phenotype, learning and memory, and one anatomic region, the hippocampus, and evaluate and manipulate the network activity? And for that, we collaborated with Zhengrong Tang and his postdoc Xuang Hao, where they can do deep brain stimulation following the same parameters used in the clinic for deep brain stimulation, stimulating the fornix that has projections onto the hippocampus and activate the hippocampal region. Specifically, they can record in the dentate gyrus and see this activation. And they did that in symptomatic Rett mice, applying an hour of deep brain stimulation for a day for 2 weeks, and then assessed for all the hippocampal phenotype, the learning and memory, the plasticity, neurogenesis, and we did the gene expression studies. And again, using what neurosurgeons will use in patients, Parkinson and other. And I'm just going to show you an example here in a cartoon. We're using a learning and memory paradigm where the animal is in a pool, where they have to find the platform to get out of the pool by remembering the cues that are on the wall of the room. You'll see that the Rett mice normally will keep on swimming and have a hard time locating the platform. But after deep brain stimulation, they do that well. I'm not showing all the data because they've been published, but for Nacl, deep brain stimulation improved all the features of Rett that are hippocampal mediated. Hippocampal learning and memory, the LTP normalized, neurogenesis normalized, and many of the changes in gene expression normalized. So this told us that the Rett brain, at least in mice, is responsive to neuromodulation and inspire us to look at other brain regions now that the Tang lab is doing to see would it help motor function. But more importantly, the Tang lab, both Xuang Ha and Qi Wang in his lab, looked at other disorders because deep brain stimulation is agnostic of the gene. It could help other disorders that may share phenotypes. And they did discover that for Nacl, deep brain stimulation also improved hippocampal plasticity and normalized feed-forward inhibition in female mice, modeling another neurodevelopmental disorder, CDKL5 deficiency disorder. So this tells us then, for many of these disorders, maybe the loss of function of these critical genes is dampening the function of the neurons because the neurons are still there. So perhaps circuit manipulation can be helpful. Now, I did share with you that Rett has many, many symptoms. And while we knew that you can do deep brain stimulation, we wanted to know, would it be possible to mimic the effect of deep brain stimulation by perhaps doing intensive training? And we wondered if the timing of the training would be critical. The reason we wanted to try this, because Rett has many, many symptoms, and it's going to be really challenging to treat it with deep brain stimulation alone, because you could do maybe the hippocampus and motor, but there's so many other features. So we wanted to see if a less invasive training would work. And this is work of Nate Akili, who was a graduate student in the lab, where he first wanted to see if there's an effect on motor. If you put the Rett mice on a rotating rod, they will fall. But we wanted to see if we treat them before symptoms appear. So this is the onset of motor incoordination symptoms in Rett mice. They're fine till about 12 weeks, and they start falling off. And by 24 weeks, they cannot last at all on the rotating rod. So if you don't train them and just train them for this 4 days, they're going to fall off. They will do very poorly. So we wanted to see what happens if we start the training before symptom onset and keep doing it every 2 weeks. So it's not a lot of training, but it's every 2 weeks, and follow them till they are 24 weeks, 6 months old, and typically very impaired if untrained. But to be sure, it's just not the number of training. We also tested it after symptom onset. And the results were really clear. Here you'll see the healthy mice, they stay nicely on the rotating rod, but the naive Rett mice at 6 months fall off, very poor performance. The late training did help this motor coordination phenotype, but not as well as the early training. Here you'll see that the early training dramatically improved their performance, and we followed the animals, they continued to improve. Of course, this is not a cure, but it did delay the onset of motor coordination by months, and even when the onset happened, it was milder. So we wanted to see, could you now do another phenotype, the learning and memory? I mentioned to you, if you put the healthy mice in a pool with a hidden platform, the mice will, after 4 days of training, will quickly go to that platform, but the Rett mice have a hard time doing that. So for this test, we had to start the training earlier, because in this particular feature, it appears in the mice much earlier, 6 to 7 weeks, they start poor performance on this task. So we really only had a month to do extra training. So we did just 2 extra training sessions, but started one in the pre-symptomatic period. And then we did them squinch just towards the end before testing, and of course, the naive. And here again, we saw a big difference. If you see healthy mice, after 4 days, they reach the platform. But the Rett mice who are naive, they cannot reach the platform. And the Rett mice who were trained late, in purple here, again, performed as badly as the naive mice. However, if you start the training early, and of course, if you train wild-type mice, either early or late, they're going to do well. But in the brown here, you see the Rett mice who have been early trained, and now they can reach the platform just after 4 days. And it really shows you this early training helped them when they were tested on the platform to perform very well. And we can then challenge them by taking out the platform and see, do they remember? And of course, there are 4 corners. So 25% is chance. And you see that the wild-type animals, half of the time, they'll find where the platform used to be after you take it out, but the Rett naive don't, late training don't. However, early training helps them find the platform and perform much better closer to healthy animals. And even the amount of platform crossings, you'll see that the healthy and the early trained Rett mice do that much better. What was interesting, though, is that we learned that although this hippocampal learning and memory improved in the Morris-Water maze, it did not improve on another task. Deep brain stimulation improved both, because deep brain stimulation stimulated the whole hippocampus. But this physical training paradigm seems to have helped only the task on which the animal was trained. So this made us wonder, when you train the Rett mouse, are you really training the cells, activating the cells that are performing a specific task, and that's why the benefit? And we decided to test this by labeling those cells. And I will show you here that we have a way to label those cells using a technology called FastTrap developed by Li Chun Lu. You'll see here mice that are just handled. You don't see much red cells because they're not in the pool. These are not important for hippocampal and cortical neurons that typically are activated when the mouse is swimming in the pool. So we have a way to monitor the neurons that are involved in the task by labeling them and by expressing in them channels that can either activate or silence them. So we have this genetic trick we can do in them. And they are channels that Brian Roth initially described that can be expressed in these neurons and can respond to a drug that only these designer channels will respond to. So this channel can be silent by this drug, CNO, while this channel can be activated. And we express these channels now in the neurons that perform the task. We can ask what happens if we either silence these neurons or activate them. So here first, we express the channel that can silence the neurons that the animal was trained on. This is the control. We just put a random cherry protein that doesn't respond to the drug. And you can see after training, the rat mice do well. But if we train all the same amount, early training, and now silence those neurons that were engaged in the task, the animals perform poorly. So here again, you see a replication experiments. If you early train, rat mice perform as well as wild type animals, as you see here. But if you silence the day before you test them, those task-specific neurons, that benefit is gone. We then asked, how about if we don't do repetitive training? We just engage the neuron, label them, know which ones they are, but instead now we have an activator channel that we put express in those same neurons and just give the drug and simply put the animal back in the cage by activating these neurons. And now you see, if you simply put the animal on the task once, but activate the same neurons engaged in the task a couple of times and test again, now the rat mice do well again. So both wild type and rat mice can do well if never trained, but at least if you now activate these neurons, they both do well. So this is really was quite informative because it told us pre-symptomatic training improves motor and memory deficit in rat mice in a task-specific manner, and that discontinued training delays the onset of the disease, and the task-specific neuron mediate this benefit. And I didn't show you the data here, but we found that the neurons themselves improve. Remember, I told you they have decreased activity. This activity improved, and their synaptic function improved. This is really important and has implications for Rett syndrome. I mentioned to you that Rett syndrome, the children appear normal for the first year, sometimes up to a year and a half. It varies from girl to girl. The average age of diagnosing Rett syndrome is 3 years. So we have this long period of time if we have known that the child has Rett syndrome, and if we began perhaps training on language, on motor activities, on other functions, might we delay the onset of the disease till much better therapies are in place? And even when we have therapies in place, this early training might make them most responsive to therapy. So I think we hope that with time, this newborn screening can help us test this hypothesis. When we have children born with Rett, if we engage them in early training, might we have a better outcome and improve this disease? And in the last 5 minutes, I'd just like to come back to the MECP2 duplication syndrome. I mentioned to you, we have a great mouse model that had the human gene and the mouse gene, and that reproduces all features of the syndrome. So we wanted to ask, if we normalize MECP2 level in adult symptomatic mice, can we rescue the phenotype? We did it genetically, but we wanted to also do it with a molecule that can be used therapeutically, such as antisense oligonucleotides. And antisense oligonucleotide are small pieces of DNA that will bind the RNA. And when there is a DNA-RNA heteroduplex, RNase H will degrade the RNA, so there will be less protein made. So we collaborated with Ionis, who gave us ASOs against the human gene. You remember my mice had one mouse gene and one human gene. So this degraded the human gene and normalized the level of the protein. And this is work done by Hezi Steinberg. And here you see an example that the transgenic mice are hypoactive. Their activity is normalized after the ASO. They can rear on their hind legs. They can enter the center. And here you see a single animal, healthy animal, the duplication animal less active, the duplication animal treated, normalized activity. We even wanted to wait till the animals had seizures every day. By 7 to 8 months, these mice will have seizures every day just before they die. And we wanted to try to normalize the protein level and see what happens. And what we saw while the transgenic had, again, this is work with Zhen Rong, who recorded the EEGs, you see seizures all the time, and the seizures disappeared, and clinically and electrophysiologically after ASO treatment. So to translate this to human study, Yin-Yao Xiao wanted to now provide the proof of concept data by creating a humanized mice where both alleles are human, because in humans, you have two identical alleles. You don't have one human, one mouse. So we wanted to be sure we can titer the dose, and we were able to do that. And she gave one dose of antisense oligo intraventricularly, which will mimic the intrathecal dose that could be used in human. And they've done many pharmacokinetic studies and followed various parameters in these animals. And what we learned is that within 1 week, the RNA is normalized. The protein, the MACB2 protein, normalizes within 2 weeks. And all the gene expression changes that I showed you that are altered in these animals begin to normalize by 2 weeks, but take almost up to 8 weeks to be all normal. But what was really interesting is that it takes the beginning of the 8th week and 10 weeks and beyond where we can begin to see improvement. So this is really was quite important for us to learn, because it told us that the molecular rescue happens fast. The behavioral rescue will take a while. You have to almost normalize everything, get the neurons back to a healthy state and engaged to be normal. It also told us we have an opportunity to monitor MACB2 levels and monitor some biomarkers that respond to MACB2 and know that we've selected the right dose. This way, one can titrate the second dose, either increase it or decrease it, or if one has overcorrected, because this is a dosage-sensitive protein, you can use an antisense to the antisense to immediately shut off the antisense oligo. So these are the kind of experiments that one has to do and be really careful about many of those dosage-sensitive proteins. So just to wrap up then, what I shared with you today on Rett syndrome, I hope you would agree, has implications for other childhood neurological disorders. Genes whose haploinsufficiency can cause neurodevelopmental disorders might also cause neuropsychiatric phenotype when the mutations are milder. So we need to pay attention to that, study the function of these genes and the effect of these mild mutations. It is important to establish which proteins are tightly regulated and pose dosage sensitivity challenges before trying any therapy. If you want to do gene therapy in Rett syndrome, you have to control the level of the protein, because remember, half of the cells have the normal protein, and now we know too much MACB2 can be toxic. The same if you lower MACB2 in the duplication, and I think this will apply to many other proteins. Early intervention will prove beneficial for many neurodevelopmental disorder. And I think this should be evaluated. We know from autism that early intervention makes a difference. So I think it's going to be really important to assess what we've done in the Rett mice, pre-symptomatically or early, if it'll work. And we hope that newborn screening in the future will permit early intervention and ensure equity, because I think, some people who are diagnosed later, some of them may come from more, racial populations who have the resources, who continue to pursue finding a diagnosis. But perhaps if you had newborn screening, you're sure everybody is getting a diagnosis early, and hopefully everybody will enroll in studies that will improve their outcome. For the therapeutics in child neurology, I think they're going to be moving forward to molecular as well as network-based, perhaps DNA and RNA-based therapy, such as the ASOs and gene replacement or gene activation, and even editing in the future. These are going to be at the forefront of therapeutics. The challenge is that we need better delivery system to reach all the desired brain cells. We have to scale up the production of such therapies and contain the cost so that we can treat as many people in as many countries as possible. We have to eliminate off-target effects and again, need titratable approaches to fine-tune dosing. There's also a room for network-based therapy. The one advantage of network-based therapy, it can be applied to multiple disorders that share symptoms. As I shared with you about deep brain stimulation for the fornix, there could be some cell-specific activation or other types of neuromodulation that improve outcome. And it is agnostic of the genetic cause. But of course, we need a better understanding of circuits in health and disease, and we need less invasive and more precise approaches. I should emphasize again, what we have today, such as physical, occupational, and cognitive therapy, will help and should always be implemented until more definitive therapies are in place. With that, I'd just like to thank the most important people who really have done all the beautiful work with their own hands that I shared with you. I mentioned them as I went along. Ruthie Amir cloned the Rett syndrome gene, and I talked about people as I went through their experiments, but a team of people have really taught us so much about Rett syndrome, and another team is currently working on that, and our collaborators in computational biology, the Zhendong Liu's team, Jianrong Tang for the physiology and deep brain stimulation, and our collaborators at Ionis Pharmaceuticals for the ASO studies. My greatest gratitude for the families who's really enrolled in our studies and supported us, and of course to our funders, including Howard Hughes, NIH, and the foundations, the Rett Foundations and RSRT, Cure MDS, and the Keck Foundation who took a risk on the DBS studies. And this is just a picture of the lab members, some of the faces. There are always some graduating, but you'll see some of the faces who did the work I shared with you today. Thank you very much. I'm happy to take questions.
Thank you so much, Huda, for the fantastic talk. So we now open the session to questions. If you have any question, please raise your hand and you will be unmuted to ask questions. I know that Dr. Susan Korick has a question. Susan?
Yes, thank you. That was an amazing talk. I learned a lot. I'm not a genetics person, but I wondered if you could comment on what's thought to be the reason why Rett's or other developmental disorders oftentimes have a time lag before symptoms present. You know, is there something about early child development that's not as sensitive to the MECP2 deficiency? You know, like why are the kids apparently relatively normal in the first year and a half of life, and then why does that change?
Excellent question. There are two reasons for that we now know. One reason is molecular. The MECP2 protein binds methylated cytosines. And there are two types of methylated cytosines. The CG is when a cytosine followed by guanine, which are throughout the genome, and the CAs, when it's followed by an adenine. And it's also throughout the genome. And it turns out MECP2 prefers binding to the methylated CA, and that binding to methylated CA is what's driving many of the features of Rett. We've done studies towards this end, and many other investigators have done studies that showed that MECP2 bind to that, and that this is a driver for these phenotypes. Now, when does the CA mark come? It's late post-developmentally. So the DNMT3A that writes this DNA mark does this postnatally in the mice, typically up to 5 to 8 weeks perhaps. It's totally written, and in human, it goes on in early childhood. and happens also postnatal. So that's one factor, but that's not the whole answer, because we've done another experiment where we asked, if we now let the animals grow with the normal protein and take out the MACP2 gene in adults, do we now see features immediately? Because of course you're having mature brain, no more critical period, all of that, right? The animals learned everything. To our surprise, we found when you knock it out in the adult, there's also a lag time till the symptoms appear. So this tells us the lag time is probably, just as I shared with you, it took a time for the recovery in the MACP2 duplication when we treated with ASO because the neurons had to recover. It takes a time for the neuron to sort of degrade their activities. So many molecular changes happen, so many physiological changes happen, and only when this happens and happens in enough neurons, you're going to begin to see features. So I think it's a combination of the necessity for the protein postnatally, because the key mark is postnatal, as well as because somehow it takes a long time for the neuronal dysfunction to happen.
Thank you. Any other question? I know that Dr. Mariam Bousohail, she asked question in chat as well.
Yes, thank you, Dr. Huda, for this amazing presentation. Want to ask regarding males with severe encephalopathy and found to have CP2 gene mutation.
Yes, so males are rare. Most of Rett syndrome happens due to a mutation in a sperm. So majority of Rett girls, 95% probably, or a little bit more, have the mutation from a mutation in his sperm, sporadic mutation. So the father is healthy, just one sperm will have this mutation. And we see it in females because the father gives the one of the X chromosomes to the daughters. Because fathers don't give an X to a boy, they always give the Y, that frequency of mutation then is much lower. Males have rarely had mutations because it has to come from the mother, and mutations in the mother are much rarer than mutations in the male. This is just because of how the DNA modified in male sperm. So the rare cases of males come from a mother who's either a carrier of a mutation, but she's not symptomatic because of X inactivation patterns protecting her, or because it's in her oocyte. And those rare males, unfortunately, the males that have non-functional protein, because they don't have another X to protect them, like the females where they have 50% of the cells, the X, are going to be very severe. And the majority of males that totally lack a functional protein die in infancy. But as I mentioned, there are males out there that have milder mutations. And I think those are more of those are yet to be discovered. I asked, there's a question in the chat from Christina Skrimpiak. What is the genomic explanation for exercise training? That's really a great question. What you mean is what happens at the gene expression level. And Yan Lee, a graduate student in our lab, is trying to answer this question right now, Christina. What she's doing, we're repeating the whole experiment I shared with you. Early training, late training. You remember we can capture these neurons. She's going to capture these neurons and ask, what's different in the Rett neurons early on versus later on? And what happened to the Rett neurons with early training versus late training? What gene expression changes happen? What's driving that improvement? So we hope to have an answer for that.
Yeah, thank you so much for your answer. I was thinking that it's something like that, kind of an adaptive process. Right. It's like the genomics knowing that soon something will happen and it's trying to adapt.
Correct. So we think it's happening at the gene expression level. We'll learn that.
Thank you so much.
Dr. Feldman?
Yes, thank you. Great talk. Enjoyed it tremendously. I was wondering how far are we from clinical trials in humans with MACP2 mutations?
So they're different, of course. For the antisense trials that I know most about. They are in clinical readiness phase, preparing for clinical trials. So we hope it's going to happen, you know, in the near future. This is work really happening between the physicians at Texas Children's and other hospitals, as well as Ionis Pharmaceuticals. For the gene therapy trials, there are now efforts to try to test new therapies with gene therapy vectors that control the level of the protein. And I believe those are also in clinical readiness phase and preparing. So I am hoping in the next 2 years or so, we will begin to see some of these trials ongoing. As you know, there's always safety and other issues to be considered and biomarkers in place, and we have to assess. I think one thing we learned, at least from animal studies, it's important to really follow a long period. Short-term follow-up may not reveal all the effects you might see from a trial, but I think this is really hopeful. And I think we need to continue to press on with different modalities that down the line will hopefully be less invasive.
Amazing, thank you.
Any other question? Okay, I take the liberty to ask a question, Huda, as well. So thank you for the fantastic talk. So I wanted to know that what happens to a histone modification, specifically with ASO therapy? Do you see like any change there as well?
That's a great question. We have really focused mostly on the gene expression changes in the ASO therapy, so we corrected that. But to answer your question, we've actually, Samir Bajikar, a postdoc in our lab, is just wrapping up an experiment right now where he did knock down the protein and in a time-dependent manner assessed multiple histone mark to really find what happens at the gene level. In the past, we've seen certain histone marks that's changed in either gain or loss of MACP2, and the histone marks will go the opposite way. But what we're trying to correlate now is specific histone marks that may correlate with the gene expression changes. Particularly, are there those that are upregulated? Is there increase in acetylation? And does that happen coincident with the gene expression? So we are collecting these data now, and hopefully we will be able to have them soon. I saw Mister raised her hand.
Yes.
Oh, hi. So thank you for this really exciting talk. It's just extraordinary how the discovery of a diseased gene can has taken us from this that moment to where we are now. So thank you. I'm wondering if you could tell us whether there are, about current efforts to implement newborn screening.
So great question, Beth. And just as a historical background, Beth was the editor at Nature Genetics when we submitted our paper, and she did a heroic job to get it in a month. So I'm really grateful for that. So what we've learned is it's a complicated topic. So many people have to really be on board to do that. Maybe the best thing to do is to do it in a contained way. Take one of those developmental disorders that are a little bit more common, where you can do pilot study and where you have data that warrant a clinical study. So there have been conversations with the NIH to think about that. But the Simons Foundation, through the Safari initiatives, is also really interested in that. And they held a meeting on that. And I think they are contemplating perhaps doing a focused study where you can pick one or two, or maybe disorders where you do that in a very planned way, in a clinical study way, where you follow the children, enroll them in clinical trials of intervention to really show that there is a benefit, as we've seen in the mice. And if you show that, then it warrants early diagnosis and newborn screening. So that work is ongoing, Beth.
Thank you very much.
Jihong has a question. Jihong, go
John,
ahead.
I asked you to unmute yourself so you can.
Oh, sorry. It's very wonderful talk. And as you know, in the rare onset disease, not only early onset, the Rett syndrome, the Alzheimer, the APP duplication mutation, and Parkinson and alpha-synuclein duplication mutations. And those mutation is not causing specific disease. It is causing the same disease in other cause of the mutation, like in presenting mutation, APP mutation causing Alzheimer. So what I'm saying is how those late onset mutation duplication gene doses changing, and how we think that's how we should approach that as a therapeutic point or cause of disease in the versus developmental disease just have one gene causing disease.
So let me simplify your question, Jihong, just a little bit for everybody else. I think what Jihong is saying is you talked about neurodevelopmental disorder and dosage sensitivity and so on, but in neurodegenerative disorders, we also have dosage sensitivity. Synuclein, APP, tau, all these proteins showed us that. And I would add ataxin-1. We know a slight increase in ataxin-1 due to the polyglutamine expansion causes toxicity, whereas loss of function, as you and Rudy and our collaboration revealed, makes you vulnerable to AD pathology, right? So I think, Jay, what you said is true, but there are different types of proteins. Perhaps if a protein is more critical for synaptic function, you're going to see earlier phenotype, neurodevelopmental phenotype, very early on with either changing its dosage in either way. If a protein is important, but not necessarily for that synaptic functions, for the neuron to function at full capacity. However, with age, its accumulation somehow stresses certain cellular machinery, either the mitochondria or the lysosome or the proteasome. That will then have secondary effect and cause toxicity in the cells. So the two are actually dosage sensitive proteins driving disease, but in different, perhaps due to different mechanism. I wouldn't be surprised if some of the disorders that affect synaptic function in their milder form, they may cause later on neurodegenerative diseases. That's remained to be seen.
Okay, thank you.
And I know that Lynn has a question as well. Lynn?
Thank you, Huda. That was wonderful as always. I just want to clarify that for the current public health newborn screening across the world, the current criteria most use the Wilson-Younger criteria, which requires a treatment.
Right.
There are many of us, in fact, I'm co-editing with Olaf Bodemer a special issue for genomics and ethical issues for genomics in the newborn period. We're trying to expand the criteria, but because of the economic factors, we have to be careful of weighing one for the other and at the same time being an equalizer to have social justice issues. Any thoughts that you have how to implement it would be wonderful.
Great question. Thank you for asking it. Let me add very clearly, that's exactly what we should do, Lynn. It has to be shown to be effective. All we have to go by now is we have benefit, we have proof of concept, it's effective in the animal models. The data are very clear from early versus late. The question is, if we did this study, we have to do a research study where, let's say, if we did it for Rett syndrome and we do for 3 years, a trial where we do this intensive training, and you don't even have to do really the control study because you have so much data from the natural history study on Rett syndrome, you will quickly know if that early intervention is making a difference. And only then, if we show that early intervention really makes a difference, if we can delay the disease onset by 2 to 3 years, we can now get more out of the girls, if they can, if they can still walk more straight, if they can use their hands, if they can say 2 or 3 words and retain those words longer until other therapies. So to me, we have to first do step 1, a clinical trial. We do the newborns in a contained study way where you do a trial and show that this intervention over 3 years is making a difference. And if we show that, then that should fulfill your criteria that newborn screening for disease X can be used because it's going to change the outcome, it's going to improve the outcome, it's going to delay the onset. It is a treatment.
Okay, thank you everyone. Dr. Zoghbi, thank you so much again for accepting our invitation and for the fantastic talk. We conclude today's event here, and I invite you to attend our next lecture, which will be on May 17th by Tracy Johnson from UCLA. Thank you everyone for your time, and we're looking forward to seeing you in the next lecture. Thank you.
Thank you so much. It was a pleasure. Bye-bye.